Edited by Michael S. Gazzaniga, University of California. Kirsty J. Whitaker. Adolescence. Adolescence is associated with genomically. Kirsty J. Whitaker HTT. Kirsty J. Research article. Neuroscience. Adolescence is associated with genomically patterned consolidation of the hubs of the human brain connectome. Kirsty J. Whitaker https://orchid.org/0001-8099KW4001@cam.ac.uk Petra E. Verts https://orchid.org/0002-0992-3210 Rafael Romero Garcia +16 and Edward T. Bullmore https://orchid.org/0002-8955-8283 Authors, info, and affiliations. Edited by Michael S. Gazzaniga, University of California, Santa Barbara, California, and approved May 26, 2016, received for review February 16, 2016. July 25, 2016. 113, 32, 9105-9110. https://doi.org/10-1073/PNAS 1601745113 36 436361 PDF/EPUB. Significance. Abstract. Results. Discussion. Methods. Acknowledgments. Supporting information. References. Significance. Adolescence is a period of human brain growth and high incidence of mental health disorders. Here, we show consistently in 2 MRI cohorts that human brain changes in adolescence were concentrated on the more densely connected hubs of the connectome, i.e., association cortical regions that mediated efficient connectivity throughout the human brain structural network. Hubs were less myelinated at 14Y, but had faster rates of myelination and cortical shrinkage in the 14 to 24Y period. This topologically focused process of cortical consolidation was associated with expression of genes enriched for normal synaptic and myelin-related processes and risk of schizophrenia. Consolidation of anatomical network hubs could be important for normal and clinically disordered adolescent brain development. Abstract. How does human brain structure mature during adolescence? We used MRI to measure cortical thickness and intracortical myelination in 297 population volunteers aged 14 to 24Y old. We found and replicated that association cortical areas were thicker and less myelinated than primary cortical areas at 14Y. However, association cortex had faster rates of shrinkage and myelination over the course of adolescence. Age-related increases in cortical myelination were maximized approximately at the internal layer of projection neurons. Adolescent cortical myelination and shrinkage were coupled and specifically associated with a dorsoventrally patterned gene expression profile enriched for synaptic, oligodendroglial, and schizophrenia-related genes. Topologically efficient and biologically expensive hubs of the brain anatomical network had greater rates of shrinkage/myelination and were associated with overexpression of the same transcriptional profile as cortical consolidation. We conclude that normative human brain maturation involves a genetically patterned process of consolidating anatomical network hubs. We argue that developmental variation of this consolidation process may be relevant both to normal cognitive and behavioral changes and the high incidence of schizophrenia during human brain adolescence. Sign up for PNAS alerts. Get alerts for new articles or get an alert when an article is cited. Manage alerts. Adolescence is associated with major behavioral, social, and sexual changes as well as increased risk for many psychiatric disorders. 1. However, human brain maturation during adolescence is not yet so well understood. Historically, pioneering studies used histological techniques to show that distinct areas of cortex were differentially myelinated in post-mortem examination of perinatal tissue, suggesting myelinogenesis as an important process in human brain development. 2. 3. MRI can measure human brain development more comprehensively and over a wider age range than is possible for post-mortem anatomists. The thickness of human cortex can be reliably and replicably measured by MRI. 4. And longitudinal studies have shown that cortical thickness, CT, millimeters, monotonically shrinks over the course of postnatal development, with variable shrinkage rates estimated for different age ranges. 5 to 11. Review in Ref. 12. CT typically shrinks from about 3.5 millimeters at age 13 Y-old 9 to about 2.2 millimeters at age 75 Y-old. 10. 11. Rates of cortical shrinkage are faster during adolescence, approximately minus 0.05 mm/Y, than in later adulthood or earlier childhood. 9. What does this MRI phenomenon of cortical shrinkage represent at a cellular level? There are broadly two tenable models, pruning and myelination. Basic physical principles of MRI predict that shorter longitudinal T1 relaxation times reflect either a reduction in the fraction of watery cytoplasmic material, like cell bodies, synapses, or extracellular fluid, or an increase in the fraction of fatty, myelinated material, like axons. Pruning models propose that cortical shrinkage in adolescence represents loss or remodeling of synapses, dendrites, or cell bodies. 13. Myelination models propose that the cortex appears to shrink because of an increasing proportion of myelinated axons, without necessarily implying any loss or change of neuronal material. 5. In the macaque monkey, although the main phase of synaptic pruning and neuronal loss occurs earlier in development. 14. 15. There is evidence for further synaptic remodeling during adolescence. 16. 17. In rodents, there is histological evidence for increasing intracortical myelination during adolescence, especially at the deeper cytoarchitectonic layers of cortex, V and 6, 18, 19. At a cellular level, this signal likely reflects a developmentally late myelination of efferent axonal segments immediately distal to the axonal hillock of pyramidal cells. 20. MRI sequences have been recently developed for myelin mapping in humans. 21. These techniques include methods based on magnetization transfer, MT, which have been validated as an MRI marker of myelination by post-mortem imaging, and histological studies of multiple sclerosis, a demyelinating disorder. 22. Details are in methods. It has been shown that primary sensory and motor areas of cortex are more heavily myelinated than association cortical areas. That most cortical areas show progressive increases in myelination over the course of normative development into middle age, and that myelination is concentrated in deeper layers of cortex. 21, 23, 24. Association cortical areas have been identified among the highly connected hubs of structural brain networks. 25. Suggesting that differential rates of intracortical myelination might be related to differences between regions in their topological roles as part of the connectome. Developmental changes in structural and functional MRI network topology have been reported. Review in Ref. 26, but not previously related to measures of cortical shrinkage or myelination. Recently, several studies have linked brain regional gene expression to axonal connectivity in the mouse, 27, 28, and functional MRI networks in humans, 29, 30. But there have been no previous efforts to investigate genetic mechanisms of adolescent myelination of human cortex. In this context, we aim to test three hypotheses. 1. That adolescent cortical shrinkage was coupled to intracortical myelination. 2. That adolescent cortical shrinkage/myelination, also known as consolidation, was concentrated anatomically on association cortex and topologically on the most strongly connected regions, hubs, of the human brain anatomical network, and 3. That adolescent consolidation of these connectome hubs was associated with a specific gene expression profile, enriched for neuronal and oligodendroglial function, and enriched for risk of schizophrenia, a neurodevelopmental disorder with its highest incidence in young adults. We used quantitative multiparameter mapping, MPM, 31, to test these hypotheses on MRI data acquired from a sample of 297 healthy young people sampled from primary healthcare registers stratified by age, and balanced for sex in the adolescent age range 14 to 24 Y-old, with tilde operator 60 participants in each of 5 age-defined strata, 14 to 15 Y-old inclusive, 16 to 17 Y-old, 18 to 19 Y-old, 20 to 21 Y-old, and 22 to 24 Y-old, methods and SI appendix, table S1. From the MRI data, we measured CT, millimeters, and MT percentage units, PU, at each of 308 cortical regions for all participants. We used linear models to estimate baseline CT and MT at 14 Y-old and age-related rates of change in the period from 14 to 24 Y-old, delta CT and delta MT, from data on participants of all ages at each regional node. We explored the relationships between these local cortical MRI markers and a few key metrics of complex network topology that have been widely used in prior neuroimaging and other neuroscience studies. Review in Ref. 32. We focused on the degree and closeness of each node as measures of nodal hubness and the community structure of the network, defined as a set of sparsely interconnected modules. Details of topological connectome analysis are in Methods and SI Appendix. We investigated the relationships between gene transcriptional profiles and colocalized MRI, CT, and MT, and network topological phenotypes by multivariate analysis of MRI data on 297 adolescents and whole genome gene expression maps of 6 adult human brains postmortem, provided by the Allen Institute for Brain Science, ABS 33. Details are in Methods and SI Appendix. For robustness and generalizability, we first analyzed data from a discovery cohort, N equals 100, balanced for age and sex as per the sample design, and then replicated all of the key findings in a validation cohort, N equals 197. Non-identifiable data and all analyses codes are available at the Neuroscience in Psychiatry Network Cortical Myelination Figshare repository. Details are in SI Appendix. Results: CT and shrinkage. CT at 14Y of age ranged between 1.93 and 3.8 millimeters across different cortical areas. Baseline CT was thinnest in primary somatosensory and visual cortices, Figure 1A, 34. In the adolescent period from 14 to 24 Y-old, there was evidence of significant cortical shrinkage, R2 equals 0.10, P equals 0.006, estimated global rate of shrinkage. Delta CT equals minus 0.011 mm/Y, Figure 1B. Although 289 of 308 nodes exhibited cortical shrinkage, delta CT 0, only 79 nodes showed shrinkage that was statistically significant at p less than 0.05 after false discovery rate. FDR, correction for multiple comparisons, Figure 1C. The regions with the greatest rates of shrinkage were located in association cortex, intracortical MT, and myelination. By combining data from neighboring voxels in each regional node, we could estimate regional MT for each depth at sub-millimeter resolution, 4, corresponding approximately to different layers of cortex, Figure 2A. MT was estimated at 10 fractional depths of the cortical ribbon from the PL surface, 0% depth, to the gray/white matter, GM/WM, boundary, 100% depth, and 2 infracortical locations 0.4 and 0.8 millimeters below the GM/WM boundary. At each cortical depth, we estimated baseline MT at age 14 Y-old and adolescent change in MT over the age range of 14 to 24 Y-old. In all regions, as expected, baseline MT increased monotonically as a function of increasing depth from the PL surface, reflecting greater density of myelinated fibers in deeper cortical layers, Figure 2b and movie S1. However, there was marked regional variability. Figure 1c areas of primary sensory cortex had greater MT at age 14 Y-old than association cortical areas. The rate of adolescent change in MT, delta MT, was greatest when measured at 70% of the depth of cortex, Figure 2D and movie S2, i.e. tilde operator 1.9 mm below the PL surface and tilde operator 0.8 mm above the boundary with white matter across all cortical regions, R2 equals 0.17, P less than 0.001. Delta MT equals 4.98 times 10⁻³ Y-1, Figure 1D. 70% cortical depth corresponds approximately to the level of lamina V and 6, comprising the internal layer of pyramidal or projection neurons, Figure 2C. There was marked regional variability in the rates of myelination. Regions with the greatest rates of myelination were located in association cortices, Figure 1G. In light of these complementary anatomical and developmental profiles of CT and MT, it is not surprising that there was a strong negative correlation between baseline CT and MT, R2 equals 0.43, P less than 0.001, beta equals minus 0.120 pu/mm. Figure 1F, and a strong negative correlation between rates of cortical shrinkage and intracortical myelination. R2 equals 0.22, P less than 0.001, beta equals minus 0.126 pu/mm. Figure 1H. Adolescent cortical shrinkage was significantly but not entirely mediated by age-dependent changes in MT, SI appendix, Fig. S1E, indicating that myelination is necessary but not sufficient to account for cortical shrinkage. Internal replication. All of the results reported so far were based on analysis of the discovery cohort, N equals 100, 20 in each of 5 age bins, 50 females, and all were closely replicated in the validation cohort, N equals 197. Tilde operator 40 in each of 5 age bins, 98 females, SI appendix, FIGs, S1 and S2. Because the validation cohort had twice as many participants, it conferred greater statistical power to test hypotheses. We henceforth focus on the most precise estimates of CT and MT at 70% cortical depth, estimated from the total sample, N equals 297, 60 per age bin, 30 females. Although all additional analyses were also reproduced separately for the discovery and validation cohorts, SI appendix, FIGs, S3 and S4. Genomic patterning of adolescent cortical consolidation. To elucidate the molecular mechanisms of local change in CT and MT, we explored the association between these MRI markers and regional gene expression profiles from human adult brain microarray datasets provided by the ABS 33. For example, baseline MT was correlated with regional expression of the gene for myelin basic protein, MBP, across cortex, Figure 2E, confirming that the MT signal is indicative of myelin content, 22. The association was strongest at 70% cortical depth, R2 equals 0.21, P less than 0.001, beta equals 0.0723, SI appendix, Fig, S3A. to explore analogous associations between all 4 cortical MRI metrics and all 20,737 genes measured in the ABS microarrays at each of 308 regions. We used the multivariate dimension-reducing technique of partial least squares, PLS 35. This analysis defined a few PLS components, which were the linear combinations of the weighted gene expression scores, predictor variables, that were most strongly correlated with one or more of the MRI markers' response variables. CT at 14Y, MT at 14Y, delta CT, and delta MT. The top 2 PLS components explain 28% of the variance in the MRI response variables permutation test, p less than 0.001. The first partial least squares component, p less 1, figure 3a, represented a significant association between a rostrocaudally patterned gene expression profile and baseline measures of CT and MT at 14Y-old. Figure 3b and C and SI appendix, Fig. S5. The second independent partial least squares component, PLS2, figure 3d, represented a significant association between a dorsoventrally patterned gene expression profile and measures of adolescent cortical shrinkage and myelination. Figure 3e and F. Dataset S1 has a full list of significantly over or underexpressed genes represented by the first 2 PLS components. Focusing on the gene expression profile defined by PLS2 because it was specifically associated with adolescent cortical shrinkage and intracortical myelination. We found that this transcriptional signature was significantly enriched in genes relating to synaptic transmission, p less than 0.001, regulation of glutamatergic signaling, p less than 0.001, and potassium ion channels, p less than 0.001, 36, SI appendix, Fig. S3G. We also found that the transcriptional profile associated with association cortical consolidation was significantly enriched for an oligodendroglial gene set, p less than 0.001, 37, SI appendix, Fig. S3F, as well as a set of genes robustly associated with risk for schizophrenia, a neurodevelopmental disorder, p less than 0.001, 38, SI appendix, Fig. S3G. Adolescent cortical consolidation and the connectome. We used the measurements of CT on the total sample to estimate the mean structural covariance matrix, representing the pairwise correlation of CT between each possible pair of 308 regions. This matrix was threshold to construct a binary graph or structural covariance network that had a complex topology, consistent with many prior reports of human anatomical connectomes, SI appendix, Fig. S639. Globally, the network was small world with hubs, indicated by a fat-tailed degree distribution, a modular community structure, and a rich club or core of densely interconnected high-degree nodes or hubs, which were located primarily in frontal and parietal association cortices, figure 4a and SI appendix, Fig. S6. At a nodal level of analysis, high-degree hub nodes also had high closeness centrality, indicating short path length of connections to other nodes in the network, figure 4b, as well as long physical distance of connections. Cortical shrinkage was faster for topologically central hubs with long-distance connections, figure 4c, degree, R2 equals 0.14, p less than 0.001. Beta equals minus 1.48 times 10 minus 3 mm minus 1. Closeness, R2 equals 0.18, p less than 0.001. Beta equals minus 4.91 mm minus 1. Distance, R2 equals 0.09, p less than 0.001. Beta equals minus 0.81 times 10 minus 3. Intracortical myelination rates were also faster for long-distance hubs, figure 4d, degree, R2 equals 0.07, p less than 0.001. Beta is equal to 3.81 times 103 pu minus 1. Closeness. R2 equals 0.13, p less than 0.001. Beta equals 14.8 pu minus 1. Distance. R2 equals 0.21. P less than 0.001. Beta is equal to 4.42 times 10. 3 millimeters per pu. We found that degree, closeness, and connection distance were also positively correlated with the pattern of gene expression associated with adolescent change in cortical structure, p-2, degree, R2 equals 0.21, p less than 0.001, beta equals 178, closeness, R2 equals 0.26, p less than 0.001, beta equals 572 times 10 minus 3. Distance. R2 equals 0.15, p less than 0.001. Beta equals 100 millimeters. Figure 4e. In contrast, these metrics were not so strongly correlated with the pattern of gene expression associated with cortical structure at 14-year-old, p-1, degree, R2 less than 0.01, p equals 0.904, beta equals minus 2.36, closeness, R2 less than 0.01, p equals 0.892, beta equals 9.39 times 10 minus 3, distance, R2 equals 0.02, p equals 0.02, beta equals minus 35.2 millimeters. Discussion. We have shown that adolescent cortical shrinkage is related to changes in intracortical MT in humans. This observation supports myelination models of shrinkage and, more generally, confirms association cortical myelinogenesis as a key neurodevelopmental process in adolescence. Our baseline MT-based myelination maps, Figures 1 and 2, show a high degree of correspondence with previous myelin maps based on alternative MRI parameters, 23, 24, 40 to 42. Across modalities, the highest levels of myelin are located in areas of primary isocortex comprising many large pyramidal cells, which are the principal targets and sources of axonal projections, 43. However, arguably, the strongest evidence that MT is representative of myelin content in these data is that MT was highly correlated with regional expression of the gene for MBP and that the set of genes most strongly correlated with adolescent change in MT was significantly enriched for oligodendroglial-related genes. We interpret our findings as indicating that adolescent cortical myelination was greatest in association areas, which were least myelinated at age 14-year-old. It is plausible, although not directly shown by these data, that specialized motor and sensory cortices may show faster rates of intracortical MT change compared with association cortex at earlier stages of development. This prediction would be compatible with the idea, dating back to Flechsig, 2, that association cortical areas are the focus of a relatively late wave of myelinogenesis. The intracortical location of strongest maturational changes in MT, corresponding approximately to the boundary between layers V, internal pyramid, and 6, further suggests that adolescent intracortical myelination may be concentrated on the proximal segments of efferent projections from pyramidal neurons, 20. Although adolescent intracortical myelination was greatest in areas that showed fastest rates of cortical shrinkage, it did not entirely explain age-related changes in CT, likewise. Although the gene expression profile associated with adolescent change was enriched for genes related to oligodendroglial function, it was also significantly enriched for neuronal genes, especially those implicated in remodeling of synapses or transport of glutamate-containing vesicles. Thus, It seems plausible that genetically programmed processes of synaptic remodeling act together with locally coupled processes of intracortical myelination to consolidate synaptic and axonal connectivity of association cortical areas in adolescence. The set of gene transcription markers most strongly associated with this late maturational process, 44, was enriched for genes known to confer risk for schizophrenia, 38. Generating the hypothesis that deviation from the normative developmental trajectory of cortical hub consolidation could be an intermediate phenotype underlying the high incidence of the clinical phenotype of schizophrenia in young people. However, we note some important caveats that mandate additional critical testing of these results and their hypothetical implications. First, the gene expression profiles used in this analysis were measured in 6 post-mortem adult brains, mean age equals 43-year-old. Brain regional gene expression is known to change somewhat over the 2 decades approximately intervening between the oldest subject in the MRI dataset and the average age of the subjects in the genomic dataset, 45. Any such age-related changes in gene expression will have confounded our analysis of the association between imaging and genomic variables. We might expect this age disparity to reduce the statistical power to detect true MRI/mRNA associations rather than inflate the probability of false positive associations. Nonetheless, the genomic analysis of developmental MRI phenotypes would certainly be stronger in the future if it was informed by age-matched regional gene expression profiles. Such data are not currently available. Second, the MRI changes that we have interpreted developmentally are estimated from cross-sectional data. A longitudinal design incorporating multiple repeated MRI scans over time would provide a more secure basis for estimation of age-related changes attributable to the developmental maturation of individual brains. Third, we have not presented any evidence that people at risk for schizophrenia do, indeed, show abnormal cortical hub consolidation, although we note that there is prior evidence for abnormal CT, MT, and cortical shrinkage in patients with schizophrenia. 46, 47. Future longitudinal studies of network development, and ideally, also gene expression. In young people genetically or environmentally at risk for schizophrenia will be required to test the pathogenic role of hub consolidation more definitively. It is well known that brain networks generally have complex topological properties, including hubs, modules, and rich clubs, 39, 48. We replicated these findings by graph theoretical analysis of structural covariance networks derived from the CT measurements, showing that topologically central nodes had higher rates of adolescent shrinkage and myelination and higher levels of gene expression associated with cortical consolidation. In a sense, this result is not surprising given our earlier data showing that the coupled changes in CT and MT were greatest in association cortex and prior studies showing that network hubs of the structural brain connectome are disproportionately located in association cortex, 39. It suggests that adolescent cortical consolidation is topologically targeted to optimize the performance of network hubs. For example, synaptic remodeling and intracortical myelination of cortical hubs might be expected to minimize the conduction time for axonal propagation of electrical signals or enhance the synchronization of oscillations across anatomically distributed cortical areas. 49. These physiological implications of connectome hub consolidation would hypothetically favor more integrative functional network topology and dynamics, which are known to be important for later maturing, higher order, cognitive functions, 50. We predict that age-related cortical consolidation of human cortical network hubs will prove to be relevant to the normal acquisition of cognitive and behavioral skills during the adolescent transition from childhood to adulthood and may also prove to be implicated in the clinical emergence of neurodevelopmental and psychiatric disorders, such as schizophrenia, in young people. 1, 3. Methods. Design. In total, 2,135 healthy young people in the age range 14 to 24 Y old were recruited from schools, colleges, National Health Service, NHS, primary care services, and via direct advertisement in North London and Cambridgeshire. To populate the secondary MRI cohort, 300 participants were subsampled from the primary cohort, conserving sex and ethnicity balance, with N equals 60 in each of 5 age strata. Additional detail and exclusion criteria are in SI Appendix. Participants provided informed written consent for each aspect of the study, and parental consent was obtained for those aged 14 to 15 Y old. The study was ethically approved by the National Research Ethics Service and conducted in accordance with NHS research governance standards. MRI data acquisition. All scans were acquired using the MPM sequence 31, implemented on 3 identical 3T whole-body MRI systems, Magnetom TIM Trio, VB17 software version. Siemens Healthcare, 2 located in Cambridge and 1 located in London. Between site reliability and tolerability of all MRI procedures were satisfactorily assessed by a pilot study of 5 healthy volunteers each scanned for tilde-operator 25 min at each site, 31. The between-site bias was less than 3%, and the between-site coefficient of variation was less than 8% for both longitudinal relaxation rate, R1, and MT parameters, 31. R1 and MT were quantified in MATLAB, the MathWorks Incorporated, using SBM8, www.fil.ionucl.ac, UK/SBM, and custom tools. Details are in SI Appendix. Estimation of regional CT and MT. The cortical surface for each participant was reconstructed from the R1 image by the following steps: skull stripping, 51. Segmentation of cortical gray and white matter, 52. Separation of the 2 hemispheres and subcortical structures, 52 to 54, and finally, construction of a smooth representation of the gray/white interface and the PL surface, 52. We use FreeSurfer version 5.3.0 software to implement these processes, Surford, nmr.mgh.harvard.edu. After quality control, 3 participants had to be excluded from the analyses because of movement artifacts, which prevented accurate surface reconstructions. We used a backtracking algorithm, 55, to parcellate 66 regions defined by sulcogyral criteria in the DesiGAN, Kiliani Atlas, 56, into 308 contiguous parcels of approximately equal area, 500 mm2, across both hemispheres in standard space, SI Appendix Fig. S2a. This parcellation template was then transformed to the native space of each participant's image to minimize geometric deformation of the MRI data and prevent age-related bias. 57. Before estimation of CT of each region. To localize cortical myelination at a spatial resolution corresponding approximately to the scale of 6 lamina of neocortex at each regional node. We estimated MT at each of 11 equidistant points on the normal line from the GM/WM boundary. Fractional cortical depth equals 100% to the PL surface, 0%. By measuring MT at depths defined in proportion to CT, we aim to adjust the measurements for variation in absolute CT between different regions and as a function of age. We additionally estimated MT at 2 locations, consistently defined as 0.4 and 0.8 millimeters below the GM/WM boundary, to sample the dense myelination of central white matter as a point of comparison for intracortical MT. Details are in Figure 2 and SI Appendix. Gene expression dataset. Microarray data for 5 male and 1 female donors with a mean age of 42.5 Y old were available from the ABS 33 human.brain-map.org. We matched the centroids of the regions of the MRI parcellation to the closest regional gene expression profile. Microarray data were averaged across all samples from all donors in the matching anatomical region across both hemispheres. The data were also averaged across probes corresponding to the same gene, excluding probes that were not matched to gene symbols in the AABS data. 2 MRI regions were excluded because both the mean and the range of gene expression values in these regions were outliers compared with the other cortical regions of interest. The final output was a matrix of z-scored expression values for each of 20,737 genes estimated in 306 MRI regions. Details are in SI appendix. Gene expression data for individual genes, such as MVP, were obtained from the appropriate row of this matrix. Similarly, gene expression values for larger gene sets of interest were extracted from the whole genome data. These hypothetically motivated gene sets included 94 genes that were identified as oligodendrocyte-specific by Cahoe et al. 37 and 349 protein-coding genes recently identified as schizophrenia risk genes, 38, of which 312 were successfully matched to genes in the ABS dataset. Structural covariance and network analyses. The structural covariance matrix comprised the pairwise correlations of CT for all possible pairs of regions in the parcellation template. Binary graphs were constructed to be node-connected, with a connection density of 10%, 39. From this graphical model of the connectome, we estimated 2 measures of topological centrality at each node, degree and closeness. These metrics are representative of the more general concept of nodal centrality, and they are strongly correlated. We also estimated the connection distance as the mean of the Euclidean distance of all of the non-zero edges at each node. On average over all nodes, we estimated clustering and global efficiency and combined these measures of network segregation and integration, respectively, to estimate the small worldness of the global network, 58. The community structure of the graph was nearly decomposed by maximization of modularity, 59, into a set of sparsely interconnected modules or subgraphs. The core/periphery structure of the graph was defined as a small rich club or core of highly interconnected hubs embedded in a larger set of more peripheral nodes. Details are in SI appendix. Statistical analyses. At each regional node, we fitted a simple linear regression model across all participants to estimate the gradient, delta CT and delta MT, and intercept or baseline, CT and MT, for each MRI measurement. For MT, the same model was fitted separately at each depth level between the PL surface and 0.8 mm below the GM/WM boundary. The null hypothesis of zero age-related change in delta CT or delta MT was tested globally on average over all regions and at each region individually, controlling the FDR at 5% to correct for multiple comparisons entailed by regional analysis. PLS regression on the gene expression matrix was used to identify the linear combinations of genes that best predicted the response variables, CT, MT, increment CT, and increment MT. The statistical significance of the goodness of fit of the first 2 PLS components was tested with two-tailed alpha equals 0.05 by 1,000 permutations of the response variables. The error in estimation of the weight of each gene on each PLS component was assessed by bootstrapping, and the ratio of the weight of each gene to its bootstrap SE was used to rank the genes according to their contribution to each PLS component. We used gene ontology enrichment analysis tools to identify and summarize annotations corresponding to biological processes that were significantly overrepresented, FDR equals 5%. Details are in SI appendix. We predicted hypothetically that the transcriptional profile strongly associated with adolescent cortical consolidation, PLS2, should be enriched for genes associated with oligodendroglia, 37, or risk for schizophrenia, 38. We tested for significant enrichment by a permutation test of the normalized bootstrap weight of each gene in PLS1 and PLS2 summed over all genes in the set. Acknowledgments. We thank Dr. Amy Orsporn for her graphic design of Figure 2C and Dr. Fred Dick for helpful discussions of MRI analyses. We also thank the AIBS for use of the Allen Human Brain Atlas available from human-brain-map.org. We use the Darwin supercomputer of the University of Cambridge high-performance computing service provided by Dell Incorporated and funded by Higher Education Funding Council for England and Science and Technology Facilities Council. Study data were collected and managed using REDCap electronic data capture tools hosted at the University of Cambridge. This study was supported by the Neuroscience in Psychiatry Network, a strategic award by the Wellcome Trust to the University of Cambridge and University College London, 095844/Z/11/C. Additional support was provided by the National Institute for Health Research Cambridge Biomedical Research Center and the Medical Research Council, MRC/Wellcome Trust Behavioral and Clinical Neuroscience Institute. PV is supported by the MRC Grant MR/K020706/1. EVI is supported by the Gates Cambridge Trust. MM is supported by the Biomedical Research Council. Supporting information. Supporting information, PDF. Supporting information. Download. For 100.81 kilobytes. Appendix, PDF. Supporting information. Download. 2.18 megabytes. NOS.16017451113.SD01 XLSX. Download. 2.61 megabytes. NOS.16017451113 SM01 AVI. Download. 590.86 kilobytes. NOS.16017451113.SM02.AVI. Download. 505.48 kilobytes. References. 1. Teephouse, M. Keshvan, J. and Jid. Why do many psychiatric disorders emerge during adolescence? Nat Rev Neurosci 9, 947 to 957, 2008. Crossref. PubMed. Google Scholar. 2. P. Fletchig, Developmental, Myelogenetic, Localization of the Cerebral Cortex in the Human Subject. Lancet 158, 1027 to 1030, 1901. Crossref. Google Scholar. 3. D.J. Miller, et al., Prolonged Myelination in Human Neocortical Evolution. Prognatal Academic Sci. USA 109, 16480 to 16485, 2012. Crossref. PubMed. Google Scholar. Show all references. View full text download PDF. Further reading in this issue. Research article July 25, 2016. Cell size and fat content of dietary restricted Cenorhabditis elegans are regulated by ATX2, an MTR repressor. Daniel Z. Barr. Shaky Chair. Josef Gruenbaum. Research article July 25, 2016. Kin of co-authorship in 5 decades of health science literature. Mattia Prosperi. Ian Buchan. Vettelai, Torvik. Research article July 25, 2016. Unraveling the drivers of MERS-CoV transmission. Simon Kochemes. Pierre Nivelet. Neil M. Ferguson. Trending. Research article March 3, 2025. Brain aging shows nonlinear transitions, suggesting a midlife critical window for metabolic intervention. Age-related cognitive decline is associated with metabolic, vascular, and inflammatory changes, making it challenging to distinguish primary causes from secondary, downstream, effects. This study demonstrates that brain aging follows A. Understanding the key drivers of brain aging is essential for effective prevention and treatment of neurodegenerative diseases. Here, we integrate human brain and physiological data to investigate underlying mechanisms. Functional MRI analyses across 4. Botond B. Ontal. Helena van Nieuwenhuizen. Lillian R. Mujica-Parody. Research article December 30, 2013. Bodily maps of emotions. Emotions coordinate our behavior and physiological states during survival salient events and pleasurable interactions. Even though we are often consciously aware of our current emotional state, such as anger or happiness, the mechanisms giving emotions are often felt in the body, and somatosensory feedback has been proposed to trigger conscious emotional experiences. Here we reveal maps of bodily sensations associated with different emotions using a unique topographical self-report method. In Lori Namenma. Enrico Glorian. Yari K. Hayatanen. Research article June 11, 2018. Neural network retuning and neural predictors of learning success associated with cello training. In sophisticated auditory, motor learning such as musical instrument learning, little is understood about how brain plasticity develops over time and how the related individual variability is reflected in the neural architecture. In A, the auditory and motor neural systems are closely intertwined, enabling people to carry out tasks such as playing a musical instrument whose mapping between action and sound is extremely sophisticated. While the dorsal auditory stream has been shown to Indiana Woolman, Virginia Penhune, Robert J. Zator. Now reading. Adolescence is associated with genomically patterned consolidation of the hubs of the human brain connectome. PDF/EPUB. Sign up for the PNAS Highlights newsletter. Get in-depth science stories sent to your inbox twice a month. Subscribe. Browse. Current issue. PNAS Nexus. Special features. List of issues. Collected papers. PNAS in the news. Front matter. Journal club. Multimedia. Podcasts. Information. About. Sustainable Development Goals. Editorial board. Authors. Reviewers. Subscribers. Librarians. Press. Cozzarelli Prize. PNAS updates. Copyright 2025 National Academy of Sciences. All rights reserved. Online ISSN 1091 to 6490. PNAS is a partner of Chorus, Clocks, Cope, Crossref, Orchid, and Research for Life. Contact. Site map. Terms and privacy policy. Accessibility. PDF help.